Hey everyone, and welcome to today's Explainer. If you are a ninth grade student joining us right now, you are in exactly the right place. Get ready because we're about to go on a seriously fascinating journey. We're going to travel all the way from the everyday limits of what human eyes can see down into the microscopic, totally invisible world of cells. It's literally an incredible hidden universe right under our noses, and today we're going to break down exactly how scientists manage to uncover its secrets. So here is our game plan for today. First, we'll talk about the limits of human sight. Then, we'll dive into Robert Hooke's famous discovery, followed by the actual magic of magnification. After that, we'll compare light and electron microscopes, measure an onion cell together, and wrap the whole thing up with a quick recap.
All right, let's jump right into Section 1, the limits of human sight.
Now, you might be wondering, why can't we just look at our hands and see cells? It's a great question. Well, think about this. Imagine taking a pen and drawing two tiny dots right next to each other on a piece of paper. If you start moving those dots closer and closer and closer together, eventually there comes a breaking point where your eyes just can't tell them apart anymore. They totally blur into one single point. And you know what that exact breaking point is? It's exactly 0.1 millimeters. When you're looking at something from a normal reading distance, which is about 25 centimeters, if 2 points are closer together than 0.1 millimeters, your eyes simply cannot see them as separate things. No way around it. In science, we actually have a specific name for this. It's called the limit of resolution. Basically, it's the ability of the human eye to see two very close objects as separate and distinct. And like we said, our biological limit is 0.1 millimeters, which is the same as 100 micrometers. Now, here is where we run into a major problem. Most cells are only between 10 and 100 micrometers wide. Because they fall completely under our limit of resolution, they're basically invisible to us unless we use a little technological help.
Moving right along to Section 2, Hooke's famous discovery.
Enter an English scientist named Robert Hooke. Way back in the year 1665, Hooke designed his very own microscope. It was pretty advanced for its time, capable of magnifying things about 200 to 300 times. Well, he decided to examine a super thin slice of cork under his new invention, and what he saw literally changed biology forever. He noticed these tiny little box-like compartments. They actually reminded him of the small, simple rooms that monks lived in, so he famously named them cells, a name that, as you definitely know, stuck around.
And you know, the whole journey of how we see the microscopic world is so cool. We start with the unaided eye, totally stuck at that 0.1 millimeter limit. Then we use the light microscope, kind of like the one Hooke used, to finally break past that barrier and see actual cells measuring between 1 and 100 micrometers. And finally, pushing the boundaries of science today, we have the incredibly powerful electron microscope that lets us see right down to the tiny organelles inside the cells, measuring just 1 nanometer or more.
Which brings us to Section 3, understanding magnification.
Let's take a closer look at the classic tool you're actually going to use in your school lab, the light microscope. To make super tiny objects appear larger, this microscope uses curved pieces of glass called convex lenses. And it doesn't just use one, it uses a combination of them. Right up at the top, you have the eyepiece. That's the part you look directly through. Then, down near the stage where your slide goes, you have the objective lens, which sits just above your specimen.
So how do these lenses actually work together to make things look bigger? The math is incredibly simple. To find your total magnification, all you have to do is multiply the power of the eyepiece by the power of the objective lens. That's it. For example, if you're looking through a standard 10x eyepiece and you've clicked a 10x objective lens into place, 10 times 10 gives you a total magnification of 100x. This means whatever you're looking at appears 100 times larger than its actual real-world size.
All right, Section 4, light versus electron microscopes.
Let's compare our everyday lab microscopes with the really heavy hitters. A light microscope, just like the one we've been talking about, uses visible light to illuminate whatever you're looking at. It's absolutely fantastic for seeing the general outline and structure of whole cells, usually in that 1 to 100 micrometer range. But an electron microscope? Wow, that is a completely different ballgame. Instead of light, it actually uses a concentrated beam of electrons. Because electrons have much, much shorter wavelengths than visible light, this incredible tech allows scientists to see intricate details down to the nanometer scale.
Just take a look at this setup. What you're seeing is a modern electron microscope hooked directly up to a digital monitor. Over the decades, scientists have constantly worked to improve 3 really important features of these machines. First is resolution, which is how clear the image is. Second is contrast, or the difference in brightness. And third, of course, is magnification.
Giant machines like this represent the absolute pinnacle of that technology. They've turned microscopes into incredibly powerful diagnostic tools that can literally show us the building blocks of life.
Okay, let's put all this theory into practice in Section 5, measuring an onion cell. Imagine you're right there at your lab bench. How do you measure something you can barely see? Well, first, you place a clear plastic ruler under the microscope to measure the diameter of your field of view. That's basically the circle of light you see when you look in. Let's say it measures 5 millimeters across. Step 2, we need to convert that to micrometers. Since there are 1,000 micrometers in a single millimeter, 5 millimeters equals 5,000 micrometers. Make sense? Okay, step 3, swap the ruler for your onion peel slide and just count how many cells fit in a straight line straight across that 5,000 micrometers circle. Let's say you count exactly 25 cells. Finally, step 4, simple division. You divide the total field by the number of cells. And boom, here is our result. 5,000 divided by 25 equals 200. This tells us that the actual real-life size of one single onion cell in your specific sample is exactly 200 micrometers wide. Pretty clever, right?
But here is where the magic of magnification really blows your mind. You just calculated that the cell is 200 micrometers in reality. But remember that 100x total magnification from earlier? If you look at that 200 micrometer cell through a 100x microscope, it appears 100 times larger to your eye. So 200 times 100 is 20,000 micrometers. And you know what that converts to? That is exactly 2 centimeters. Thanks to those lenses, a practically invisible speck suddenly looks the size of a small coin right there in your field of view.
All right, we made it to Section 6, recap and final thought. Let's lock in everything you need to remember for your next exam or lab session. Number 1, the human eye's limit of resolution is 0.1 millimeters, which is exactly why we need microscopes in the first place. Number 2, Robert Hooke used an early microscope to discover and name cells back in 1665. Number 3, you can always find total magnification by multiplying the eyepiece power by the objective lens power. And finally, number 4, to estimate the size of a cell, simply divide your field of view in micrometers by the number of cells you can count across it. Before we wrap up, I want to leave you with this one final thought. Think about it. A simple, single drop of pond water holds an entire, complex, totally invisible universe buzzing with life that you could never ever see with your bare eyes. But with a microscope, that barrier is completely gone. So as you head into your next science class, ask yourself, what hidden worlds are you going to discover next? Thanks so much for joining me on today's Explainer. And hey, happy exploring!